Yeah, thank you so much for the introduction. My name is Alejandro. I'm a postdoc at Princeton. And yeah, today I'm really excited to be here to present you our kind of like recent work. So thank you so much for the organizer for putting these together. And as you can see, it has to do with bacterial collective migration via chemotaxis, which I will explain later, and death. So the two only certain things in life, death and chemotaxis. But so here in the part of death in this work, it has to do with bacteriophages, as I just mentioned. So bacteriophages, or phages for short, these are viruses that infect and kill bacteria. So phages are the most lifelike entity on Earth, and they have been co-evolving for billions of years with bacteria, developing their own immune and defense mechanism against bacteria. And then bacteria, they counterattack with their own, basically. And here, just to showcase them, basically you see a bunch of phages attacking an E. coli cell, binding on the cell wall with the tail proteins. And then in this pictorial view, what you can see is that they inject their DNA material. And then after that, inside the cytoplasm, you have like the whole biosynthesis, like DNA replication, protein synthesis, and then self-assembly. So they self-replicate inside the cell wall, and then they lyse the cell, and they release a bunch of phage particles. As you can see here in this video, they lyse the cell. This is from Raju lab. And they can release up to like many hundreds of phage particles that they can infect other healthy bacteria. And in particular, in the work that I'm going to present today, I'm going to focus on the life cycle that I just mentioned, which is called the lytic phage life cycle. So which is represented in this particular cartoon here, which is what I just explained you. But there are other type of phages, like called lysogens. They can integrate the DNA into the bacterial DNA, and then they can divide. And then when conditions are good for them, they can activate this life cycle, and they can release more phage particles. So one particular question is, why do we care about these phages? So, you know, since they kill bacteria, the relationship between bacteria and phage is important for the global ecology. But as a consequence of this predation of bacteria, you know, they're a promising avenue to treat bacterial infections in different contexts, like the human health, but also in industry, or just in the environment, like for instance, like the food industry. And something important is that usually in the lab we have typically studied like these interactions between bacteriophage and their well-mixed condition, either in test tubes or like on other cases in just Petri dishes on agar plate, like for instance plaque acids. However, these kind of like experiments, they don't mimic the environments where they typically cohabit in nature. So you know, in nature, bacteriophage interactions occur in spatially heterogeneous and in many cases in mechanically and chemically complex environments. And I'm here just going to give you like some examples, like for instance, in hosts, like, you know, they can form biofilms, you can have like chemical gradients, and other contexts, like for instance in the environment, like for instance in large bodies of water. And some particular interest that we have is also in soil-like environments, so the ground beneath our feet, where of course you have a geometrically complex environment where you can have gradients of nutrients and other signals. You can also have pockets of phages. And something important is that in many of these environments, you know, like bacteria can move, which will be a central topic of this talk, and they can use chemotaxis just to navigate these environments. And, you know, we're interested in how space, you know, affects like the relationship between bacteria and phages. So bacterial chemotaxis inherently relies in spatial structure, which will be an important component of the talk. So something important, as I mentioned, is that in these environments, they use their motility and metabolism to navigate large distances. So I will show you one particular experiment from the lab. So what you're seeing here is an inoculum of E. coli cells. This is a C projection of confocal microscopy, and they are inoculated in a soil-like environment. It's a hydrogel granular material that I will explain later, but I wanted to showcase just like the fundamental mechanism that is going to appear in this talk like many times, which is collective bacterial chemotaxis. So this is very zoom out. 1 pixel is roughly 1 cell. And we do confocal microscopy, and we just observe, basically. And if you let them run, what they form is, I hope that you can see well here, they form like this stable and smooth chemotactic fronts that they run over many bodies in space, and they just coordinate their behavior collectively, basically. So the way they do that is basically through their metabolism. So since you have many cells, they're able like to consume the nutrient. They will generate a, hopefully displaying, so they will generate a local gradient, and then each cell is able to sense the gradient, and they can bias their motion into regions where the nutrients are higher. And collectively, they just coordinate to form like this localized band and colonize new terrain where the nutrients are higher and the conditions are more favorable. So one particular question that we wanted to understand is like, what happens if now we introduce phages in this kind of like soil-like environment and we challenge the bacteria? Are they going to be able to form a front, or maybe they're not even able to form it because the phages predate on the bacteria? So first, so that's kind of like the central question, like how do phages influence the dynamics of collective bacterial chemotaxis? And first, I would like to acknowledge the team. So I've been working in the labs of Sujit Datta at Princeton and Ned Wingreen also at Princeton, and in particular with Ned, we have been working on the theory side of the project, and I've been collaborating with a former postdoc who's starting her own lab in Delaware, former postdoc in Sujit's lab, Victoria Muir, who has been leading the experiment I'm going to show you today. So basically what we do in the lab, we use this particular material to mimic like this like more mechanically and heterogeneous environment, which are these granular hydrogel matrices, which we use as a platform to observe just microbes, and in this particular case to study bacteriophage interactions. So these are hydrogel nanoparticles swelling with just your favorite media for bacterial growth. And once they are swollen, they form a spherical packing. And basically they are cross-linked, like these particles are cross-linked, so the microbes cannot go inside, and also like the phages cannot as well. So basically the microbes are just sitting between the pores. And basically then, as I have shown you before in this experiment, we do confocal microscopy. And something good about this is that they are transparent. So we really can spy on the cells and just look at how they behave. So you're just seeing a motile E. coli moving around in this complex environment. Here also like tagging the flagella. And they also, they're swelling with media, so they have lots of nutrients, and they're also permeable to oxygen, which are small molecules basically. And something important for this talk is that we can tune the pore size just by the amount of nanoparticles that we put, so we can control the degree of confinement. So here in this particular paper, in others, you have like, if you want to see the details, I recommend you to go there basically, but that's kind of like the general idea. Then basically what we're going to use is confocal microscopy to visualize the real-time dynamics of motile E. coli encountering lytic T4 phages, which I just mentioned that we are going to just focusing on lytic T4 phages. So these are like the main experiments of this work. I'll walk you through. So on the top, basically you have a control case. So as in the previous experiment that I showed you, basically we have a bacterial inoculum here that is going to run into phage-free space. And in this other case, now this is dispersed with, as in the cartoon that you observed before, with phages homogeneously. So you have a dispersion of phages here of like 10 to the 8 phages per ml. So we wanted to know, okay, are they going to be able to form a front? So we ran the experiment, and just for comparison, you will see that these fronts are still able to traverse these long distances, even in the presence of phages, under like this phage-rich environment, or at least relatively rich. So a naive thought, a naive experiment is like, okay, let's keep increasing the concentration. There should be a point where we will be able like to collapse the population, and hopefully maybe the front will not form. So we ran the same experiment just by increasing one order of magnitude the phage concentration. So if you play the experiments, I'll play all of them at the same time. At some point, these are just probably lye cells, but if you wait long enough, eventually a form is still able to form, and it traverses with the same steady state speed as there were no phages, but there is a delay time of like just a few hours. So we decided, okay, we cannot collapse the population, but there should be a limit. Let's try to even increase it more. At this point, we are limited by just the amount of phages, so going beyond this is like pretty tough. So if we do that, I'll run the experiments again at the same time. So the signal here is probably cells that are lysine, so no form fronts, but we decided to wait long enough. So if you squint your eyes and you wait long enough, eventually you see that a front is actually able to form even in these highly rich environments rich in phages. So now the delay time is longer, it's like tens of hours, but they're still able to traverse it at some point with the same steady state speed. So they maintain unperturbed by the presence of phages. So that's kind of like the main punchline of the observations. Like once formed, like these chemotactic fronts can traverse like phage-rich terrains unperturbed. And we wanted to understand that. That's what's like basically the puzzle. And basically we can quantify these. So here where you can see is like the front position over time. These like for the control case, no phages, the low phage concentrations, you can see that they're on top, like the same steady state speed. And then as you increase the concentration, you will see that there is a delay time, basically, but they will, they will persist with the same constant steady state speed. Of course, then you can quantify over many replicates. You can see that the steady state speed remains unaffected, and then like just the delay time in front formation increases with the amount of phages that you have in your environment. So the question is like, how can bacteria achieve these in these challenging environments for them? And that's one immediate thought that we had is like maybe bacteria can mutate quite rapidly, and maybe they're challenged by phages. So does the evolution of phage resistance play a role in these chemotactic fronts when they have phages? So that's one of the first things that we did. Basically, once the front is formed, we decided to isolate cells from the front and then just plate them. So on the left column, what you just have is the control, like the plates don't have phages, and here is the plates with phages in kind of like a phage-rich environment, and you will see that no colonies form, so indicating that the cells are still in the front, they are still susceptible to phages, and only when they have been challenged with 10 to the 10 phages per ml, you have like some indication of like 20% of the cells are resistant. So clearly, they, at least for the low phage concentration, they do not rely on just like becoming phage resistant. So, and something important also for what I'm going to understand this and I'm going to explain you now is that, from the cells that they didn't encounter any phages, they were not exposed. Only roughly 1% or below 1% of the population of these E. coli cells are resistant to these particular like T4 phages. So, basically, we wanted to understand that, and then what we did is we turned to like modeling. So we decided to develop a minimal biophysical model with still has like some of the key ingredients of the experiments to model like bacterial chemotaxis and phage predation. So I'll try to walk you through the model. It's kind of like a standard in the field, and I'm happy to talk afterwards about the details. But essentially, what we are going to consider is like two subpopulations of bacteria. One is like fully susceptible to phages, and the other one is the 1% I was telling you that is still susceptible, but less susceptible, and there is a kind of like transition probability or mutation rate, if you want to call it, between these two, but it's pretty small. And then both populations can diffuse, and then they can bias their motion and chemotax to gradients of nutrients, as you see in this pictorial view. And then phages, they are just like passive particles that diffuse around, which is just this term here. And the two subpopulations, they can grow at maximal rate. They can consume the nutrients for growth, and that's basically modulated by this function here for the nutrients. And then they can be infected at a gamma rate, basically. And then for the phages, they absorb to the cells, infect them, they can diffuse. And then, of course, you have to take into account the infection of both populations, which eventually, after roughly 20 to 40 minutes, they will die, basically, and give rise to a burst of phages with size beta, which is the hundreds of phage particles that I was mentioning before. And then you need to also like take into account like the nutrients and basically use for growth, like your carbon sources and the chemoattractants that in this case is serine. So this is like it may seem a bit complex, but there are many things that we can measure. And the important thing is that there are two actually key parameters that control if you will have a front or not, or if this front is delaying time. One is this parameter here that compares like phage predation with the ability of like chemotaxis, like the chemotactic rate. And the other one is kind of like the MOI, so it's like the initial phage to bacteria concentration ratio. So you can run simulations basically and build a state diagram or a phage diagram, that's how we like to call it. And on the x-axis you have the MOI, and on the y-axis these other parameters I was mentioning that in particular in the experiments, we are just in this particular value here, and then we just change like the ratio of initial phage to bacteria concentration, and you can see that just by changing the phage concentration, we are able to recapitulate like the experiment. So we go from a front that it's completely unperturbed by the presence of phages to a front that is delayed but still travels with the same speed. And eventually, I mean, in the simulations, you can push further, and then you can find a boundary where you shouldn't expect like any front, because like the phages should collapse. That it's hard to access from the experiments. So these are the row of like the 3 experiments that I just showed you in the presence of phages, and basically you can, this is just a cut like through the basically you can run in the same setting as in the experiments, and then you can keep track of the bacteria, the chemoattractant, and the phages. So the phages in red. So in this particular low phage concentration, if you play the movie again, you will see that the bacteria are like quite fast, so that they kind of like they are outrunning the phages, the phage predation, and they are leaving behind a trail of phages that they just diffuse. However, as you increase the phage concentration, there is a point where they are able to collapse some of the susceptible population, and then basically the rate limiting step there is just to deplete the nutrients by the bacteria that were not killed, or they're less susceptible, and eventually traverse this phage-rich environment. And the same happens when you have a higher phage concentration. So then you can compare like the steady state speed, and you can see that in the presence of phages, they remain, the speed remains unaltered, and then due to this rate limiting step, you have a delay in front formation. And the good thing is that now we have a testable prediction for like this particular model. We can push it harder, and something that we then change is like this y-axis. So that's something that we can tune in the experiments by changing just the degree of confinement. So we can go from something that is like more constrained to less constrained, so that they can chemotax faster. And then we should expect that even in this case that there were like a delay in front formation, you shouldn't expect you should expect a conventional front. And we did experiments and also simulations, and you can see here that the front starts at the very early time, so it remains unperturbed, and you don't need you don't have a delay in front formation. And then finally, we can make further testable predictions just to test this hypothesis that you need to deplete these nutrients to keep, basically the rate limiting step is that you need a gradient, and then once you have like the front form, you can keep running. So basically we had like this thought experiment is that basically if you have a clear zone without phages, perhaps you should be able to even like, keep running in this 10 to the 10 phages per ml. So we did that, like basically the first thing is like, what does the model predict? That's what the first thing, because that's easier than running the experiment. So you will see here in red the phages, once you're formed, you're able to keep traversing even if you are this high phage concentration. So we decided to test this. So here, this is just the last experiment I'm going to show you. On top, you have the control. You have this experiment where you don't have phages here, and here you have phages, and then just for comparison, the one that I just showed you where you have like tens of hours of delay time. So you can see that, here if you form the front, you can keep running. And then compared to this, where you don't have basically a front because you have a lot of phage predation and you didn't have time to form the front, you need to wait like tens of hours. Whereas here, you're running, so you can kind of like outrun phage predation. So then you can put all together, and we found a good agreement between experiments and theory. And I'll just wrap up with these, just the main takeaway. So I hope that I have convinced you that collective chemotaxis can play a big role in helping bacterial communities escape phage predation without relying entirely on phage mutations. We have unraveled some of the key, I think, biophysical mechanism, which is, you need to form a front, and then once you're formed, like you can traverse like these phage-rich environments. And then something that we would like to go is like, what does this tell us about nature, and if there is any implications of these results for like biomedical or environmental applications. So with that, I would like to thank the team and also like the human frontier science for making this work possible. Thank you. Happy to take any questions.
Thank you, Alejandro. There is time for 2 questions.
Very nice talk. So my question is, have you done any sequencing analysis on the front group versus the one that they don't run front, and what is the changing, like the mutations or adaptation of those front group?
No, we haven't, like that's something that we want to go into that direction. We have been talking with other folks in Princeton that they do like sequencing, and I mean, it's just tough because then like you need, here we are limited just by volume, so it's tough to extract like high volumes from these, and you have like the hydrogels there. So it's more challenging in terms of like just the volume and just then removing like some of the hydrogel, but that's something that we would like to see, like what does like sequencing tell us? So far we have just done like the plate experiments that I just shown, but not sequencing. But that's a great question. Thank you.
Question? Yes, please.
Hi, I was just wondering, in the experiments, do you know if the phage migrate with the moving front, or does the front completely eliminate the phages?
We are not able to tag the phages, so it's hard to tell. I mean, we know that a lot of the GFP signals comes from just like from cells that lysed and just from bursting, but just like in these kind of like soil-like these granular hydrogel matrices, they're still like the diffusion of like the phages, which are non-motile at all. They're just passive particles. It's reduced like significantly. So we just see them as just kind of like a slowly diffusing particle. So that's the key point. Like the bacterial chemotaxis is much faster than diffusion of phages and then like this phage predation. So they kind of like escape when they have proliferated a lot. So they're just left behind. Yeah, there are actually some experiments that they, I think from Sepekuen's lab, that they have seen for other phages, like this hitchhiking effect that they can just, hitchhike on the cells. Here, like 100%, we don't know, but we expect that they're not traveling with the front. But you have proliferation, so also like cell proliferation. So the cells that die, then they are replenished. So that's kind of like the mechanism. I hope that answered the question.
A very last question.
Yeah, just a short, unrelated question, maybe. Do you know whether you can control the direction of chemotaxis by placing phages in different locations in the gel?
I mean, that would be very cool. We haven't tried that, but that's so far we have just explored homogeneous concentration of phages, but that would be very cool. Like controlling the direction, I mean, probably we could tune like the distribution of pores, and they will have like a preferential direction to go. It's tough to do in this particular setup, but that's a good point. I mean, they just go where there is more food. That's the probably you can come up with ideas of like kind of like just leading the cells in where you want to go. But that's a good point. Yeah.
Okay, so thank you very much for the very interesting presentation.
Thank you.